Hi, my name is Chantal and we'll be overviewing PFU protein purification today. The first step we're going to do is streak isolation and overnight culture, followed by a expression of IPTG and harvest with sonication. We'll also be doing immobilized metal affinity chromatography or IMAC. This is our purification step. And lastly, dialysis. When performing a streak isolation, we will use a flame, a plate, a disposable loop, and our frozen PFU sample containing the hist tag, which can be found in the -80 refrigerator. Then, we will use our disposable loop to quickly pick up some of the sample and spread it on our plate. We will dispose of our plastic loop, turn on our flame, and use a metal loop now. We will heat the loop until it turns red. Red means that it's ready. Now we will spread our sample using the side of our loop, gliding it throughout the agar in fine little lines. We will repeat this step over and over until we have 4 quadrants as seen in this demonstration. Once we successfully streak our plate and incubate it at 37 degrees Celsius overnight, we will see colonies form. The next step is to grab an inoculation needle and pick up 1 colony. This colony will be transferred into LB broth, in which we'll make an overnight culture for the next step. The next step after our overnight culture is to transfer the overnight culture into new media. Here we're using LB broth with kanamycin and chloramphenicol. These are the 2 antibiotics that are going to prevent any additional bacteria from our surroundings from growing with our desired PFU E.coli. Unfortunately, I don't have a clip of us transferring the bacteria, but normally we just gently spew it inside the flask and place it back into our incubator at 37 degrees Celsius. Here they'll be shaking until they reach an optical density of 0.6 or 0.8. We want this desired optical density because it ensures us that our PFU has grown to its maximum potential. Here is our incubator and shaker plate, and the student here is actually adding our ITPG.
IPTG, not ITPG. My goodness, Chantal.
Our ITPG is going to allow for more PFU protein expression. This is important because we'll maximize the amount of PFU purified in this step. The temperature in this incubator is switched from 37 to 16 degrees Celsius. This is key to maintain our protein from denaturing. This is the next day. We removed all our samples from the incubator placed at 16 degrees Celsius. And as you see, we have centrifuged them at max speed, and we've developed the media on top and a pellet which contains our bacteria with our PFU protein. I inserted an image on the side, and it shows my tubes without the supernatant, and each one of those tubes contains 1 gram of E. coli. Here's me attempting to decant my supernatant one-handedly. I don't recommend doing that, use 2 hands, but I'm basically eliminating my bacteria food without removing the pellet. Also, make sure that your waste cup contains bleach just in case if there's any leftover bacteria still floating in the supernatant. Here is a small clip of our centrifuge. I put it at max speed, which is 4,000 RPM. Don't do this one-handed like I did. It leads to mistakes, as you see. I had to rearrange it again because it wasn't properly balanced. A great lab safety tip here is to wait near the centrifuge until it reaches max speed. We want to make sure that everything is balanced correctly. Prior to this, I measured my samples on an analytical scale to make sure they are within the same range. I didn't get to film the sonication step, but feel free to look at Mahan's video on sonication. He goes step by step on how to do the sonication and what to expect. Usually when we do sonication, we have vibrations that open up our cell, and we can then extract our PFU from all the rest of the junk. After sonication, we're going to set up a water bath at 74 degrees Celsius for 15 minutes. This is specifically for PFU. TAC is a little bit different. After a 15-minute water bath, we will centrifuge for 30 minutes. This will allow us to separate our cell junk from our protein. In this part, I'm setting up for a gel electrophoresis, and the first step is to create the master mix. So I'm using 10x buffer, DNTP, sterile water, 50% glycerol, and my PFU extract. The total volume should be approximately 50 microliters for my protocol. After this, we're gonna use our master mix it up with some primer and some lambda DNA, and then we'll stick it in our PCR machine. In the meantime, the PCR machine will run about 2 hours, which is a long time. So I like to make my gel, and here I'm doing 2 gels. They're agarose gels, and they contain also something called gel green, which is our stain that we'll be able to see in our ChemiDot. Once the PCR is over, we want to set up our gel. Here we added SBB buffer, and we added our 2 gels. Notice that the negative and the positive is on the bottom. This is because DNA is negative, so you want it to flow down through. We also want our DNA ladder. This tells us how many base pairs. You want to be careful with pipetting. You want to make sure that you don't puncture the gel with the pipette tip, or else you could potentially risk that your samples go to buffer heaven and you'll never see them again, and it will be really sad. Also, this step takes about 45 minutes, which is a long time, and if you're like the lab mentees here, they made a little snowman in the meantime. So there are 2 methods we could go about purifying our protein. One is with a gravity column containing nickel resin. This is called IMAC, or immobilized affinity chromatography. We could also use this fancy machine called an FPLC, or fast protein liquid chromatography. And this machine, what it does to our protein is, through a series of buffers, it leads them out in a certain gradient that allows for our protein to be more purified. With immobilized affinity chromatography, or IMAC, for this part of the purification, our gravity column has nickel resin in it, and it has this bluish color. That means that it's charged. We will be working with a series of 3 buffers, and they all contain different concentrations of imidazole. If you want to see the exact concentrations and how much I'm adding, follow my protocol on PFU Protein Purification. That will go more into depth of the concentrations and our column volumes. This is just a brief overview. So we're going to add our lysis buffer, and this will equilibrate our buffers and our column with our nickel resin. Essentially, our PFU protein with our His tag will be attracted to the nickel resin, and this will cause all the flow through of the cell junk and any additional impurities to flow through the column. After we have our lysis buffer step completed, we'll move on to our wash buffer. Our wash buffer step, here we'll be adding a different buffer with a higher concentration of imidazole compared to our lysis buffer. And this will help us, again, remove any non-desired proteins with no His tags out, any cell junk. Once we do that step, we'll move on to our elution buffer. Our elution buffer has the highest concentration of imidazole. This is because we want to do a competition within our protein and whatever is in our column, so our protein can finally spew out with its his tag. Here's a quick clip of the FPLC. I don't know how to use this machine 100%. I've worked with it, but I'm pretty sure somebody else will make a full video on how to use the FPLC if you want to purify proteins or nanobodies. Once we finish purifying our PFU sample, we're going to run an SDS page. It's like a gel electrophoresis, except we're not looking at DNA amplification, we're looking at protein amplification. This will tell us which of our elutions have the most amount of protein. I won't go into depth on how the protocol is done specifically. Our elutions need to be processed before we can put them in the gel, but here is a clip of how the gel looks before it is placed into our SDS page. So we're adding our gel into this little green holder here, and then we're going to add our SDS buffer. It's kind of similar to a gel electrophoresis again. We're adding our buffer, our gel, and we're trying to get that amplification of protein. But since we're only doing 2 gels, we would fill up our SDS buffer up until the 2 gel line. And we want to also fill it inside both gels on the top. I don't know if you can see it, but it's somewhere in between the middle of those 2 gels. We want to fill that up. So we're actually running a tandem gel. That means that we have 2 gels, one on each side, and as you see here, each student is going to pipette their samples at the same time in order for us to run our samples together. This gel is actually a lot more faster than electrophoresis. It will take about 30 minutes for the gel to process at 120 volts. Here's my attempt to show you guys how the protein is going to run through the gel. It's just going to run slowly downwards. And here we are trying to remove both gels from our holder. It can be a little challenging at first removing the actual gel from our plastic, so you have to maneuver this little green tool around. Make sure you're careful with the green tool. You can actually add a little bit too much force and it can tear the gel, which is not good. A little tip I have for removing the gel from the physical container is actually just placing a little bit of DI water into like another container and then flipping my gel over, and it will just magically unstick. Here is our ChemiDoc, and this actually images our gels for our gel electrophoresis and our protein gels. The important thing here is to note what type of stain you used for each gel. So like I mentioned, for my gel electrophoresis, I used gel green, so I would pick the gel green stain on the machine. And for here, we used unstained for our SDS page, so we're going to click unstained. The scary part about this machine is actually placing the gel. You want to use like a laundry method, like Dr. K says, to gently place the gel without ripping it. Here we're just repositioning the gel, making sure it's centered, and then we're going to close it, and then we're going to take a picture of it, and then you're going to wait a long time until the machine decides to give you the image. So this is basically our last part of our protocol, which is a dialysis. We do this to remove the imidazole we added when we ran it through the column. And here's just a quick overview of how you open it. You want to put your sample in the middle part of the top with a pipette and just pipette it in. But before you do all this, you actually want to pre-soak it in your TEN buffer. That's what I used for PFU. Then you add your sample. Close it up, place it in your TEN buffer. Make sure that your TEN buffer is cool. This is so the protein doesn't denature. And when you want to remove your sample after you dialyzed it several times, you're going to use a syringe on the slides. Thanks for watching my video on PFU protein purification. And if you want to learn more about how to do each step in more detail, feel free to look at my protocol on PFU protein purification.